So now let's talk about reference sugar. So reference sugar is the sugar that we use to compare other sugars. For example, I'm having a compound glyceraldehyde. So what is aldehyde now? So aldehyde is actually a functional group. The general formula of aldehyde is C double bond O. This is alkyl and H. This is a functional group and this stands for aldehyde. And glycer means 3 carbon. So let's have a compound glyceraldehyde. C double bond O and H C C. So this is I'm having this compound is known as glyceraldehyde. Why? This is aldehyde group C double bond O H, and this is the alkyl group, and glycer because there are 3 carbons. Carbon number 1, carbon number 2, and carbon number 3. So for comparing, we will see the second last carbon. So how many carbon we are having? So here we are having 3 carbon, right? So we won't see the last carbon, we will see second last carbon. So this is the second last carbon, and we will see the functional group OH. We will always see OH, where the OH is. So here the OH is in left side. So this will be known as L-glyceraldehyde. Why? Now listen. We are having 3 carbon, right? And we have to see the second last carbon. This carbon number 2 is second last carbon, right? And we have to always see the functional group OH, like where it is positioned, where it is placed, in right or left. So in this is right side, right? This is left. We won't see the carbon number 3, we will see carbon number 2 because it is the second last carbon. So the OH is placed on the left side. So here we will write L-glyceraldehyde. For example, I'm having another compound so this is known as, so we will check 1, 2, 3. So this is aldehyde and there are 3 carbons, so this is known as glyceraldehyde, right? We will check second last carbon. We won't check last carbon, we will check second last carbon, the position of OH, right? So here OH it is there. This is second last carbon, carbon 1, carbon 2, carbon 3, last carbon, second last carbon, third last carbon. So this is second last carbon. You will see the OH where it is placed in right side, which will be known as D-glyceraldehyde. Okay, and in this way we are also having D-glucose, L-glucose, D-galactose, L-galactose. So in our body, mostly, or we can say majorly, D-glucose is present in our body. Okay, so now let's talk about optical activity. Optical activity, right? I'm having a container over here, right? And I have mixed glucose solution. So I have mixed any glucose or any sugar over here, and I am striking the light. So we will remember that the light should be polarized. Now you will say that what is polarized light? Okay, I'm telling you. When we strike the light and it goes in all directions, like in all direction, this is known as non-polarized light. It goes in many directions, like this is going. So this is known as non-polarized light. But when it moves in only in one direction, this is known as polarized light. So this is known as polarized light. So for this optical activity, we want polarized light. And now you will say that how we will convert unpolarized light into polarized light. So we have, for this purpose, we have polarimeter, right? So we are having polarimeter which converts unpolarized light into the polarized one. Okay, when we strike polarized light, either the light moves in right direction or in the left direction. So if the light moves in right direction, we will call this as dextrorotatory. Okay, and this is known as positives. But if the light moves in left one side, we will call as levorotatory. Please don't match dextro with right one and levo with this one. These both are entirely different things. We will show this with small d, whereas we were showing that with capital D. And this is small l. Okay. So why we are using this? Why we are using optical activity? What does it mean dextrorotatory? What does it mean levorotatory? When we strike light on the solution, the sugar solution, if the light moves in right direction, it means it's dextrorotatory. But if the light moves in left side, it means it's levorotatory. It doesn't mean that it has OH on the left side or right side. These both are entirely different things. Okay, but we are having a container. Okay, and it says sugar, and I am striking a light and it moves in simple path. Like it doesn't bounce, it doesn't reflect anywhere. It means it has equal number of dextrorotatory and equal number of levorotatory, right? The dextrorotatory and levorotatory are equal, and it will move in center. So this type of mixture is called a racemic mixture. So these are known as racemic mixture because it does not move in right or left or any direction. It is moving the simple straight path. Why? Because the dextrorotatory and levorotatory both are same. Now what does it show? Why we are using optical activity? What does it mean dextrorotatory? What does it mean levorotatory? Simply dextrorotatory, and like this shows presence of asymmetric carbon, and these isomers like levorotatory and dextro are known as optical isomers. So these are known as optical isomers. These shows the presence of asymmetric carbon, right? And these isomers, levorotatory and dextrorotatory, are known as optical isomers. Remember this thing that D-glyceraldehyde, L, like these, and dextrorotatory and levorotatory both are entirely different things. Like these are optical isomers, right?